In this video, we're going to take a look at a specific gene that's associated with cancer growth, in particular, focusing on breast cancer. TP53 is perhaps the most well-studied gene when it comes to cancer. It's a seemingly obscure, rather average-sized stretch of nucleotides on the short arm of chromosome 17. What does TP53 stand for? Well, the TP stands for tumor protein. So yeah, the gene makes a protein that's intricately linked to cancer formation. And what about 53? Well, that's interesting too. It was once thought that the molecular mass of the protein was 53 kilodaltons. We now know that the molecular mass of the protein is more like 43.7, but by the time that was established, the name had kind of stuck. So TP53 it is. The TP53 gene makes the tumor antigen protein P53. This P53 protein is so important to our cell biology and DNA replication processes that it's colloquially referred to as the guardian of the genome. When the DNA in a cell gets damaged, for example, from UV radiation, or from mutagenic compounds, or from bad copying during mitosis, the protein P53 acting in the nucleus can do one of two things. If the DNA can be repaired, the P53 protein can activate other genes to produce proteins needed for that DNA repair. On the other hand, if the DNA can't be repaired, then the P53 protein will prevent the cell from dividing, ultimately leading to apoptosis. To put it into context, how important the TP53 gene is when it comes to preventing cancers, more than 50% of all cancer occurrences feature a mutated version of this TP53 gene. Let's get into a little bit more detail. I already mentioned that the TP53 gene is located on chromosome 17. The gene itself has more than 20,000 nucleotides. But as with most genes, most of these nucleotides are located in intron regions. Once those introns are spliced out, the resulting mature mRNA strand has about 2,600 nucleotides in it. Then, once the 5' and 3' untranslated regions are accounted for, it ultimately codes for a protein that's 393 amino acids long. A change to any one of the coding nucleotides could change the amino acid composition of the resulting protein. So let's take a look at some examples of mutations that could occur. Okay, here we're looking at a stretch of nucleotides around the middle of the TP53 gene. Now, this is only half of the DNA helix. It's what we would call the coding strand. And right here is position 743. A single nucleotide change from a guanine to an adenine at this position would change the codon at that location from a CGG to a CAG. This means that when the corresponding protein is made, instead of coding for an arginine at that position, a glutamine is coded for instead. That tiny change decreases the effectiveness of the P53 protein and could make someone more susceptible to cancer. We would see a similar effect if there was a substitution mutation from a cytosine to a thymine at position 382, or a change from a guanine to an adenine at position 532, or a guanine to an adenine at position 818, or a guanine to a cytosine at position 841, or a guanine to a thymine substitution at position 844. You get the idea. Small, seemingly insignificant changes to this gene can change the shape of the resulting protein. A change in the protein's shape can change how well it works, and if the protein isn't being effective enough in repairing DNA or signaling for apoptosis, a cancerous growth could begin to form. Hey, did you know that cancer is exceedingly rare in elephants? Now, what does that have to do with the TP53 gene? Well, okay, you would expect cancer to be pretty common in elephants, right? I mean, the longer lived an organism is, the more prone it should be to picking up cancerous mutations on the TP53 gene. And elephants can live 60, 70, even 80 years. Given this, you would expect cancer to be common, especially amongst elderly elephants. Well, it turns out that elephants have 20 copies of the TP53 gene on their DNA. So if a cell picks up a mutation in one of its TP53 genes that makes the resulting p53 protein less effective, well, no problem. Whatever cell that is would still have 19 copies of the TP53 gene that could code for fully functioning p53 proteins. So that's the story of the TP53 gene. It's a gene that might be the most important tumor suppressor gene that we have. For other examples of specific genes that are associated with breast cancer, be sure to check out the other videos in this series or navigate over to www.evo-ed.org for teaching and learning materials, as well as other helpful resources. That's all for now. I'll see you next time.